The classification of modern humans and Neanderthals as distinct species has long been debated. Genetic evidence, morphological similarities, and patterns of interbreeding complicate this taxonomy. This research aims to evaluate whether modern humans and Neanderthals should be considered a single species under the biological species concept, which emphasizes reproductive compatibility, or other species concepts by examining genomic data, morphological traits, and archaeological evidence. The question of how strongly genetic evidence of interbreeding, morphological similarities, and cultural interactions support the classification of modern humans and Neanderthals as a single species is pivotal in understanding our evolutionary history. This research has substantial implications for how we view the evolutionary pressures that shape the development of Homo sapiens, particularly during the late Pleistocene, when both groups coexisted in Eurasia. The cognitive and cultural advancements reflected in archaeological evidence hint at shared knowledge and practices between modern humans and Neanderthals, reinforcing the idea that their coexistence was characterized by both competition and cooperation. Recognizing modern humans and Neanderthals as closely linked, if not a single species, might also reshape our understanding of species boundaries and the dynamics of human evolution, prompting us to reconsider the traditional views of the biological species concept, a theory based solely on reproductive isolation. An essential element in the discussion of human evolution is the morphological evidence that differentiates Homo sapiens from Neanderthals. Fossil records have revealed significant anatomical differences, particularly in cranial and postcranial features. However, it is important to note that our species and Neanderthals also exhibit shared traits, which complicates their classification and challenges the notion of distinct species boundaries. Genetic evidence provides insight into the extent of interbreeding between Homo sapiens and Neanderthals. Genetic studies have substantially contributed to the understanding of the relationship between Homo sapiens and Neanderthals, with evidence suggesting significant interbreeding occurring between these two hominins. The genetic component of this research will focus on sequencing high-coverage genomes of Neanderthals and modern humans. Ancient DNA will be extracted from Neanderthal fossils, while modern human samples will represent populations from diverse geographic regions to ensure broad representation. These sequences will be aligned to a reference human genome to identify shared DNA regions and calculate rates of gene flow between the two groups. Statistical tools such as d-statistics and F4 ratios will be employed to evaluate introgression patterns. Functional analyses will identify genes under positive selection in both groups, focusing on those linked to immunity, cognition, and cold adaptation. These findings will help elucidate the extent of genetic exchange and its evolutionary significance. To assess morphological similarities and differences, the study will utilize 3D scanning to create digital models of skeletal remains, including cranial, pelvic, and long bone features. Morphometric analysis will quantify these differences and explore the degree of overlap in traits such as cranial shape, brain case size, and limb proportion. Population-level variation within each group will be compared to determine whether their morphological ranges are distinct or overlapping. Functional implications of observed differences will also be investigated, particularly their potential effects on locomotion, childbirth, or dietary adaptation. Interbreeding patterns will be examined through an analysis of hybrid fossils, archaeological artifacts, and chronological data. Fossil specimens showing mixed morphological traits will be identified and dated to determine the timing and frequency of interbreeding events. Archaeological evidence, including burial practices and tool use, will provide insights into cultural exchanges between Neanderthals and modern humans. Computational models will simulate interbreeding scenarios to assess the viability and fertility of hybrid offspring. Radiocarbon dating and stratigraphy will map the temporal overlap of Neanderthals and modern humans, correlating interbreeding events with environmental or climatic shifts. By integrating genetic, morphological, and archaeological evidence, this research aims to clarify the evolutionary relationship between modern humans and Neanderthals. It has the potential to reshape our understanding of species boundaries and provide new insights into the shared evolutionary history of these two groups.